Hi everyone, I'm Lauren and myself and Natasha are going to be talking about our research surrounding the influence of the microbiome in the development of fetal alcohol spectrum disorders. So fetal alcohol spectrum disorders, or FASD, is an overarching term describing varying severities of physical, neurodevelopmental, cognitive, and behavioral deficits associated with maternal alcohol consumption during pregnancy. There are 4 diagnostic conditions which form part of FASD. Individuals with fetal alcohol syndrome present with the most severely affected phenotype. These individuals suffer persistent neurocognitive and developmental delays, musculoskeletal underdevelopment, facial dysmorphisms, and are vulnerable to psychiatric comorbidities such as anxiety and depression. Individuals exhibiting less severe dysmorphology and reduced growth retardation are likely to receive a partial fetal alcohol syndrome diagnosis, whereas individuals displaying central nervous system deficits with confirmed prenatal alcohol exposure may be described as either having alcohol-related neurodevelopmental disorder or alcohol-related birth defects. The Western Cape of South Africa has the highest prevalence of FASD, with approximately 20 to 28% of children affected, compared to the global prevalence of 0.77%. Due to the extent of physical and cognitive issues that individuals with FASD face, significant costs arise from their specialized education, rehabilitation, and healthcare. Without adequate support and intervention, those with FASD may be at increased risk of substance abuse, engaging in criminal activity, unemployment, and behavioral difficulties, including thought disorders, attention problems, and aggressive behavior. FASD therefore constitutes a serious health concern and a significant social and economic burden, particularly within impoverished and rural communities. Alcohol compromises the integrity of the intestinal barrier, allowing the maternal commensal bacteria to enter the bloodstream at permeable points in the normally impenetrable intestinal wall, and in doing so, allows the bacteria to be transported throughout the body, including to the fetus. Due to the head-first presentation of most neonates during labor, the physical contact of their oral and nasal cavities along the colonized surfaces of the birth canal facilitates the transfer of vaginal flora from mother to newborn. Exposure to these microbes results in colonization of the newborn and the acquisition of commensal intestinal bacteria. Neurodevelopment is suggested to be modulated by the gut microbiome via the microbiome gut-brain axis. This is an axis with bidirectional communication, implying that both the brain and the gut can interact and influence each other. Prenatal alcohol exposure-induced perturbations during infancy may have long-term health consequences. So abnormal gut colonization during the first weeks of life can alter crucial functions of gut microbiota, including barrier, immune, and nutritional functions, and can also increase susceptibility to disease. Furthermore, neurocognitive development in the early postnatal environment is highly dependent on microbial composition and the corresponding microbial metabolic outputs. Therefore, alcohol-induced maternal microbial alterations may result in abnormal infant gut colonization, increasing the risk of neurodevelopmental disorders in infants. Various studies have shown that stresses applied to pregnant mice induce microbial perturbation and consequently abnormal colonization of the offspring gut microbiome. These gut and vaginal microbial changes led to alterations in metabolite profiles, involved in energy balance, and also disrupted sex and region-specific amino acid profiles associated with neurodevelopment. These findings could be conceptualized in the context of FASD. So alcohol, acting as maternal stressor in excessive quantities, may lead to alterations in the gut and vaginal microbiome, resulting in abnormal microbial colonization of the neonatal gut microbiome and subsequently the neurocognitive deficits associated with FASD. Our study aims to characterize and compare the gut and vaginal microbial composition of women who gave birth to infants diagnosed with FASD and those who gave birth to infants without FASD. In addition to this, we aim to compare the gut microbial composition of infants with and without FASD. Based on the aforementioned aims, we aim to determine whether changes in the gut and vaginal microbial composition as a result of alcohol consumption during pregnancy alters the risk of FASD development in infants. So pregnant women were recruited from antenatal clinics in Robertson and Wellington in the Western Cape by the Phaser team, and audit screening was used as a primary assessment of alcohol consumption during pregnancy, And then active case ascertainment was used to make a FASD diagnosis by triangulating data from infant morphology examinations, neurodevelopmental assessments, and maternal interviews. Women were assigned to the FASD group if they gave birth to an infant or infants with FASD, or the not FASD group if their infant did not have FASD. A vaginal swab sample was self-collected under the supervision of a registered nurse in the maternity ward using the OmniGene vaginal kit on the day of birth, and then stool samples were collected from both the mother and their infants using a PSP stool collection tube. To date, we have extracted microbial DNA from vaginal swabs and stool samples. We have assessed the DNA yield and quality to evaluate microbial DNA extraction efficiency. Microbial DNA extracted from maternal and infant stool samples has been sent to the CPGR for 16S rRNA paired end sequencing of the V1, V2 region, and we have just sent the microbial DNA extracted from the vaginal swabs to the CPGR for the same sequencing. The DADA2 pipeline in R was and will be used to pre-process the FASTQ sequencing files. This pipeline is used to remove primer sequences, filter and trim FASTQ sequencing files, and construct an amplicon sequence variant table. The ribosomal database project reference database is used to assign taxonomy. PhyloSeq is used to identify compositional differences between groups, and R is used to perform statistical microbial composition analyses, as well as generate alpha and beta diversity values.
For microbiome analyses, we commonly look at two different types of diversity. Alpha diversity represents the diversity within a single sample, while beta diversity represents the diversity between samples. The maternal gut microbiome was dominated by Prevotella in purple, Bacteroides in orange, Saxonovibrio in pink, Acinetobacter in blue, and Facilobacterium in green-blue. Prevotella and Saxonovibrio are associated with plant-rich diets enriched in complex carbohydrates, fruit, and vegetables, while Bacteroides is linked to a high intake of fat and protein. Very little is known on the role of Acinetobacter. However, there has been some correlation with a protein-rich diet. Facilobacterium's sole known species, Facilobacterium prasinasi, is one of the most abundant and important commensal bacteria of the human gut. This species is an important butyrate-producing bacteria and has been considered a bioindicator of human health, as when its abundance decreases, inflammatory processes are favored. Interestingly, this genus has been shown to decrease in the third trimester of pregnancy. There were no statistically significant differences in alpha diversity between women who gave birth to an infant or infants diagnosed with FASD and those who gave birth to an infant or infants without FASD, both with prenatal alcohol exposure and without. Of the variables investigated, both trimester and Bristol stool scale had an effect on beta diversity, with an adjusted p-value following Benjamini-Hochberg correction of less than 0.05. The infant gut microbiome was dominated by Prevotella in purple, Bacteroides in coral, Bifidobacterium in orange, Facilobacterium in green-blue, and Shigella in green. In infants, bacteria in the genus Bifidobacterium digest the healthy sugars in breast milk. In fact, babies who are breastfed tend to have a high abundance of these beneficial bacteria than infants who are bottle-fed. Additionally, babies who are born by vaginal delivery usually have a high abundance in Bifidobacteria compared to infants born via C-section. Preterm birth has also been found to reduce levels of these bacteria in infants. Interestingly, species of Shigella are normal inhabitants of the human gastrointestinal tract and can cause dysentery or shigellosis. Shigella is one of the top 4 pathogens that cause moderate to severe diarrhea in African and South Asian children. There were no statistically significant differences in alpha diversity between infants with FASD and those without FASD, both with prenatal exposure and without. Of the variables investigated, Both delivery mode and Bristol stool scale had an effect on beta diversity with an adjusted p-value following Benjamini-Hochberg correction of less than 0.05. As expected, trimester had an effect on beta diversity. Major alterations in gut microbial composition occur during the third trimester of pregnancy. Estrogen and progesterone impact the composition of the gut microbiome through their effect on bacterial metabolism and result in an increased abundance of the phyla Proteobacteria and Actinobacteria and a decrease in the genus Faciliobacterium. As expected, delivery mode had an effect on beta diversity. The gut microbial composition of vaginally delivered newborns closely resembles that of their mother's vaginal microbiota. In contrast, the gut microbiome of infants born via C-section is dominated by bacterial species that are found on the skin. Bristol stool scale also had an effect on beta diversity in both the mothers and their infants. The Bristol stool scale gives an indication of transition time through the gastrointestinal tract.
After performing all of our microbiome-related bioinformatic and statistical analyses, we hope to find that the vaginal microbial composition differs between the groups of women. We hypothesized that the vaginal microbiome of women who consumed alcohol during their pregnancy and gave birth to infants with FASD will be dominated by anaerobic species of bacteria that are associated with adverse outcomes of pregnancy, and that the women who abstained from alcohol use, as well as the women who consumed alcohol during their pregnancy and had an infant without FASD, will have a stable lactobacilli-dominated vaginal microbiome. This project will produce multiple outputs, which are listed on the slide. The results generated from this project may be used in future intervention studies and can be used as a basis for future gut and vaginal microbiome research, especially within South Africa. An association between the gut and the maternal vaginal microbiome with FASD might reveal potential pathways contributing to this spectrum of disorders, which could be used for novel therapeutic interventions. The ultimate goal is that if the hypothesis is confirmed, microbe-based interventions targeted towards rectifying the gut and the maternal vaginal microbiome may be able to diminish the abnormal gut colonization of the newborn in the future, reducing the extent of neurocognitive deficits and the debilitating symptoms of individuals exposed to alcohol prenatally. We would like to thank all the people and organizations mentioned on this slide for their contributions towards making this project a reality, and the SASBi Student Symposium for affording us the opportunity to present today. Thank you.
Thank you, Lauren and Natasha. This is a beautiful presentation. I really liked all the images and the animations.
Thank you.
Voke asks, why the choice of the V1 to V2 hypervariable region? I may have missed it, but what was your sample size in the different groups?
So we chose the V1, V2 region because it's usually used for babies. So Natasha would have used it for her infant stool, and it's also used often in vaginal microbiome studies as well. And then our sample size for the vaginal microbiome is 17 in the FASD group and 10 as controls, essentially. And then I'm not 100% sure about Natasha's sample size.
So my sample is 98 women whose babies have FASD, and then 102 whose babies do not have FASD. And with the infants, it's very similar. I can't think now without looking at the slides, but it's pretty much 50-50. And then just building on Lauren's answer to the V1, V2, we have a collaborator and quite a few other people have shown that the traditional V3, 4 or V4 region doesn't represent the bacterial species as well in infant stool. So they've done various optimizations and have found that the V1 to V2 is the best.
Thank you, guys. There's another question. How is it diagnosed? Are women honest about whether they drink during pregnancy?
So obviously no. We do, or I don't do it, but the phaser team does it, an audit screening test, and they also use physiological markers to determine if they are telling the truth on their screening tools. So audit is the alcohol use identification disorders identification test, and it's basically this 10-point screening tool that's used to determine the mother's alcohol consumption behaviors, essentially. And then so that's the honesty part. We use ethyl glucuronide, excuse my pronunciation, which is in the nail clippings. And then we also use PEth, or phosphatidylethanol, to determine the alcohol consumption in the past 3 weeks in blood spots. So yeah.
Okay, great, thank you. And then George asks, are there solutions like painting the baby with a solution of microbiota post-birth?
George, do that. Lauren can answer this.
So I have read a paper about it, but they are suggesting that's probably not the best idea. There are other things like other not abnormal, but less beneficial bacterial species that may be present in the mother's vaginal microbiome, and you wouldn't necessarily want to paint that onto the baby. So I think, yeah, probably not a great idea.
But it's a hot topic.
Right.
Yeah.
Yeah, it is. It's very interesting. Microbiomes are always very fascinating. Okay, thank you so much, Lauren and Natasha. That was a really good joint presentation.
Thank you.
We now have come to the end of our symposium. Thank you to everyone who has attended, especially if you've been here for all 3 days. We will now go on to our prize giving. Natasha, would you like to take over?
Yes, I am. I'm just giving the judges a few seconds. But in the meantime, George is going to share with you a link, and we would appreciate it if you would share some feedback using that link. Yeah, so thank you so much, guys, for being here. We really appreciate it. Unfortunately, we would have really loved to see you in person, but we hope that we will see you next year, the 26th of September in Stellenbosch. And then we hope you'll stay and attend also the SASHG, not SASHG, sorry, SASBi, that was last year. Yeah, and I just would like to take some time to thank my fellow organizers, George, Matab, Noel, Laura, and Berta in her absence, for helping make this such a great symposium. I hope you guys enjoyed it as much as I did. I was very nervous, but it ended up being excellent in my opinion.
I just want to say, Natasha, thank you on behalf of the SASBi Student Council, because you really did so much. You pretty much single-handedly organized the symposium, so thank you on behalf of all of us. And you really did a great job. Just a quick note that the presentations will be available on YouTube, most likely, but we will share the link with you guys once they are all up.
Awesome, thank you. So I am nearly ready with the presentation. I apologize.
I have a question for Lauren. Lauren, who does the stool DNA extractions in your lab?
Natasha is definitely responsible for that.
Oh no.